Okay, so we're going to start with the easier divisions of the two divisions, which is the mitosis cell division. So this is going to be division of regular somatic cells. So it's the basis for growth and development of an organism, because we know that we begin life as a single cell zygote and then have to replicate that cell over and over again to become a multicellular organism. There's really two parts to the division of a cell. The division of the actual nucleus is called karyokinesis and is the more complicated of the two. And the division of the rest of the cell membrane, and the cytoplasm is called the cytokinesis. So really, if you think of a cell in terms of its time that it spends in its life cycle before it divides, the cell division part is actually a very short part. If typical cell life cycle might be 16 hours, the mitosis is only 1 hour of that 16 hours. So a larger part of the cell cycle is spent in the time between cell divisions, which is called interphase. So interphase is broken up into several stages. During that time, it carries out its normal function. So for example, if it's a beta cell on the pancreas, it's going to secrete insulin. If it is a muscle cell, it's going to make actin and myosin fibers and contract for muscle movement. So it's just going to carry out its normal functions. It's going to double in its size, and it's going to duplicate its DNA before the cell can divide. And so it's broken up into three main stages. You have the G1 phase, the S phase, and the G2 phase. So the first phase, that gap1 phase, is the pre-synthetic gap. So this is the time before the cell duplicates its DNA. And basically the cell is doing its normal thing, it's carrying out its normal functions, and it's getting ready to replicate its DNA. So it's the process of DNA replication, which we're going to go into detail later in the semester, requires a lot of different enzymes and proteins, and so all those proteins have to be manufactured by the cell before replication can occur. During the S phase, the chromosome replicates. Each chromosome is replicated. Once the chromosome is replicated, it has a different looking structure. You can see that it's doubled its DNA here. So you can see this is double the amount of DNA, but it's still considered one single chromosome. And the two, what we call sister chromatids, are held together by that central region called the centromere. Like I said, the two duplicate arms are called sister chromatids. Now during the G2 phase, that's after the DNA is replicated, and the cell is going to get ready for mitosis. So any enzymes and proteins that are necessary for mitosis, those are going to be made during that time, and the cell is going to carry out its normal metabolic functions. So the cell also continues to grow. By the end of the G2 phase, it should be double in size, and all of its organelles should be duplicated so that there'll be enough organelles for each daughter cell after division. So just one important thing to mention, to note, is that the DNA does get replicated before mitosis occurs. And that's important because each new daughter cell needs to have an equal amount of DNA. So the DNA has to be first duplicated before cell division can occur. So some cells can enter what's called the GO phase, which is the quiescent phase, because not all cells are going to continuously divide over and over again. If they did, that would be really bad, and that would lead to cancer. And so in fact, that's what cancer is. It's simply cells dividing when they're not supposed to be and forming tumors. So obviously our heart cells, our muscle cells, our lung cells don't continuously divide throughout our life. And some cells will go through periods of non-division, like our memory B cells are going to remain in the GO phase until they come in contact with their antigen that they're specific for, their pathogen that they're specific for, and then that will activate them to divide. So that will activate them to go into re-enter the G1 phase and enter the S phase. Just because they're not dividing doesn't mean that they're not metabolically active. These cells are still carrying out their functions. They're just not duplicating their DNA and dividing. So during the G1 stage, the cells have they have a decision to make. They can go on two different paths. One is they can complete G1, enter the S phase, and complete the cell cycle and divide, or they can withdraw from the cell cycle and enter that GO stage, either temporarily or forever. Now, like I said, interphase is the longest period of the cell. Mitosis only takes up about an hour. So if the cell is not actively dividing in the process of mitosis, then if you stain the cells and look at them under a microscope, you can't really see the chromosomes. The chromosomes are relaxed, they're diffuse in the nucleus, and they are in the form of chromatin. Once the cells start to enter mitosis, the first phase of mitosis, which is here listed as prophase, excuse me, that's when the chromosomes start to condense down and you can see them under a microscope. So the 4 stages are prophase, metaphase, anaphase, telophase. And we're going to go through each phase individually and describe what's occurring in the cells. So during prophase, a majority of the time of mitosis, more than half of that hour, is spent in prophase. And so during prophase, the centrioles that are a big part of assembling the spindle apparatus start to migrate to opposite ends of the poles, and they establish opposite ends of the cell and establish the poles. So the centrioles are a small part of a larger structure called the centrosome, and the centrosome is responsible for organizing those spindle fibers into the spindle apparatus. And this creates an axis in the cell that the chromosomes will eventually line up on. During prophase, also the nuclear envelope and the nucleolus are no longer visible. They disintegrate, and the chromosomes condense down into their signature structure that you can see. The 2 chromatids that make up a chromosome are held together by a protein called cohesin. So in metaphase, the beginning of metaphase, what we call prometaphase, the spindle fibers attach to the chromosomes and start to migrate them towards the center of the cell. During metaphase is when they're actually lined up in the center. So the spindle fibers connect to a specific structure on the centromere called the kinetochore, and like I said, the 2 sister chromatids are held together by a protein called cohesin. Once the chromosomes are attached to a spindle fiber and start moving to the center of the cell, there's an enzyme called separase that will break down that protein so that the arms of the chromosomes have that kind of floppy appearance because they're no longer connected to each other, but the chromosomes are still held together at the centromere because there's another protein called shigrosin that prevents the cohesin from getting broken down in that section. So you can see here, let me get something to highlight here. Okay, so here is the kinetochore, and you can see the spindle fibers attached at that region. In between the 2 sister chromatids, you can see that cohesin, and you can see where here, this part and this part here gets broken down by the separase when at the beginning of metaphase, once the chromosomes are attached to a centromere, attached to a spindle fiber. But at the area of the centromere, you have this other protein called shigrosin that's going to protect the cohesin from getting broken down so that the 2 sister chromatids will be held together at the region of the centromere. So this is a picture of metaphase. So before, the picture that we saw before was prometaphase. And so this picture is showing at metaphase you have the chromosomes lined up dead center on the metaphase plate, also called the equatorial plane. So just a little bit more about these spindle fibers. They're made up of microtubules, which is made up of the protein tubulin. They're dynamic structures that they can shorten and lengthen just by adding or removing subunits of that protein tubulin. And that's how they're able to pull and migrate the chromosomes in the direction they want them to go. The kinetochore microtubules are the ones that extend all the way from the centriole and attach to the chromosome at the kinetochore. And those are the ones that are directly responsible for the movement of the chromosomes. So once the chromosomes are lined up at the metaphase plate, anaphase can begin, and this is where the chromosomes are pulled apart. So you have the two sister chromatids that get pulled apart at the section of the centromere. So in this, by now the shigosin has to be degraded so that the cohesin can be cleaved by the separase so that the two chromosomes can be separated. That process is called disjunction. Once the chromosomes are completely separated, each individual chromosome is now referred to as a daughter chromosome. Telophase begins when the chromosomes are at their respective poles. It's the final stage of mitosis. The cytokinesis occurs. So in animal cells, it's almost as if someone ties a string around a balloon and the cell just kind of pinches off and produces what's called a cell furrow. With plant cells, because they have that cell wall, instead of pinching off, they actually just form a new cell wall called a cell plate in between the two nuclei to form two daughter cells. Also, once the chromosomes are at their respective poles and cytokinesis is occurring, simultaneously the nuclear envelope starts to reform around the chromosomes, and the chromosomes will start to relax and diffuse out to form that chromatin. Okay, this is a animation that comes with the book. If you have access to the Mastering Genetics website, it's on there. I'm also going to post this in Moodle so that you can go through this on your own. But this first slide just kind of shows you what we've already talked about interphase, how you have the G1 phase, the S phase, the G2 phase, and the GO phase are all part of interphase. And so it shows it consists of the S phase, where the cell duplicates its DNA, the GAP1, the pre-synthesis gap phase, and the G2 post-synthesis gap phase, where the before and after DNA replication. And this is showing you the plant cell during interphase, and so you can see the chromatin is all diffuse and you can't really see individual chromosomes. So this is showing once all the G1, the S, and the G2 phases are complete, then you can start the process of mitosis. So the first step is prophase, and so during prophase, the centrioles begin to organize the spindle and they move to the poles of the cell. So you can see there the spindle migration. The axis of the cell here is being determined by the positioning of these poles, of these centrioles to the poles. And in the center, the chromosomes are beginning to condense. So like it says here, although not yet easily seen, the chromosomes have been duplicated and they'll start the condensing process. So also in prophase, the nuclear envelope begins to break up, and so now you can see the chromosomes have condensed, and you can see their structure's more apparent. You can see the centromeres present in the chromosomes. By the end of prophase, you can clearly see the sister chromatids, that classic structure of the chromosomes in a dividing cell. And this is a picture down here. You can see where you can really see the chromosomes, and then all the red is the spindle fibers. So when metaphase begins, you have the centrioles have moved to the poles. They've attached to the centromeres, and they'll start actually moving those chromosomes where they're supposed to be in the center. And so by the actual metaphase, they're positioned on that equatorial plate. And this is a picture of the actual cell stained. So you can see here during metaphase, and this picture is also in your book, how the chromosomes are clearly lined up on that equatorial plane. The spindle, the pink fibers, you can see how the poles have been established in the cell, and now the chromosomes are ready to divide. So here it's showing you how those spindle fibers are actually attached to that kinetochore, which is part of the centromere. So during anaphase, the two sister chromatids are pulled apart. Once they're pulled apart, they're referred to as daughter chromosomes. And so there's a really good picture. That's also in your book. Alright, so during telophase, now you have the chromosomes are found at the opposite poles. The actual cell cytoplasm divides. That's called cytokinesis. And then a nuclear envelope starts to form around the new daughter chromosomes. So at the end of telophase, you have two daughter cells, and the chromatin starts to condense back down. The chromosomes lose their structure. And then those cells can undergo interphase again. So this is showing the same thing in plants, but note in plants, instead of the cell membrane pinching off, an actual cell plate forms between the two cells. Alright, so like I said, this animation, I'm going to have it on Moodle so that you can go through it and actually read the slides, and you can go through it over and over again on your own time. Okay, so the cycle of the cell undergoes regulation. There's several points to check and make sure that the cell is in good condition before it divides, so that mistakes aren't perpetuated to daughter cells. There's these molecules called cyclin that bind to these other enzymes called kinases to create molecules called cyclin-dependent kinases. And kinases are enzymes that phosphorylate other proteins. And by phosphorylate, I mean they just add a phosphate group. And usually it's some form of activation. So a kinase will come along, it'll pop a phosphate on a protein, and it'll activate that protein to go do something else. And that's just a big part of how cell signaling pathways work. There's always some kind of kinase that's putting a phosphate on something that then gets activated and then go and does it something else. The checkpoints, we call it checkpoints in the cell cycle, are there to make sure that the cell hasn't been damaged in some way, that if the cell were to continue dividing, that the daughter cells would be damaged as well. And that's the sort of thing that can lead to cancer. So what will happen is the cell will repair itself before it moves on to the next stage, or if it can't, it will kill itself. It will self-terminate by a process called apoptosis. So we know that there are at least 3 checkpoints in the cell cycle, and that we know that we've seen where mutations in the genes associated with these checkpoints can lead to cancers, various cancers. So this is just a figure that shows the 3 different types of checkpoints and what's going on at each checkpoint. And on the next slide, I'll explain in more detail what's going on at each checkpoint. There's another one of the little animations that comes with the book. This will be the last part of the animation will be the last part of this lecture. So then you can move on and answer the review questions, and then move on to the next stage of replication, or the next stage of the lecture, which is meiosis. Okay, so this is just another one of the animations provided by the book. That's just kind of a review of what we've been talking about the cell cycle. So what's interesting about the cell cycle is it's pretty much the same for all animal cells as far as the different phases and how it's regulated. And so, evolutionarily speaking, this regulation must be really important for it to be conserved across, all these different species. And we know that when something goes wrong with this regulation is when we end up with cells that divide uncontrollably, and so that's cancer. And so we're very interested in how the cell prevents uncontrollable division from occurring. So as far as the timeline for the cell cycle, if you look at a typical cell that has about 16-hour dividing time, from the end of one division to the end of the next division is about 16 hours. And of course that's going to be different for different types of cells and different species. But just as an example, if you have a cell whose life cycle is 16 hours, a majority of that time is spent in interphase. In fact, 15 of the 16 hours is spent in interphase, and the rest, only an hour, is spent in mitosis. And so this is showing you the G1 about 5 hours, about 7 hours in the S phase. It takes a long time to replicate all the DNA. And then about 3 hours in the G2 phase. And then we move into just an hour that we spend in mitosis. Now of that hour, a majority of that time is spent actually in prophase. So if you look at it, the first 36 minutes, so over half of the time is spent in prophase, just getting ready to divide. And then the other phases only take a few minutes each, with telophase being the second longest time. So throughout this whole 16-hour life cycle of the cell, there are several checkpoints that occur that prevent the cell from continuing further if there's some reason that it shouldn't, if there's some kind of damage that has occurred to the cell. Now, a lot of cells don't continue dividing. We've talked about this already. They can arrest in that GO stage. Some cells may arrest temporarily. Some cells may arrest in GO and never divide. You always hear the story about brain cells. Once you lose your brain cells, you can't replace them because your nerve cells in your brain, they stay in the GO phase until you die. So, and we know obviously cancer cells don't enter the GO phase, and that's a big part of the problem. So the first checkpoint is the G1S checkpoint. This is a checkpoint that occurs before synthesis. And the second checkpoint is the G2M checkpoint that occurs after synthesis but before mitosis. And then the M checkpoint occurs during mitosis. So the first checkpoint, the G1S checkpoint, is just making sure that the DNA hasn't incurred some kind of damage that needs to be repaired before it's replicated. The G2 checkpoint occurs after synthesis, and that's really important because errors can be made when the DNA replicates, and we'll talk about that more when we get to that point. But if the DNA is damaged at any point during replication, that damage needs to be repaired before the cell divides, otherwise those changes to the sequence will get set in stone, and that's what you don't want to happen. So, because then you have mutations, and that can lead to cancer. And then the M checkpoint is just making sure that the chromosomes are aligned properly, that they're all attached to the spindle apparatus properly, that the cell division is going to carry out correctly. So as review, the G1S makes sure that the cells have grown to the proper size and that the DNA is not damaged. The G2M checkpoint is making sure that the DNA was replicated correctly and that there aren't any mistakes. And the M checkpoint makes sure that the spindle apparatus or the spindle assembly has been assembled properly and that the spindle fibers are attached to the chromosomes correctly. And this is just showing you all the different cycle times and the checkpoints and everything in review. Alright, so that is it for that animation. And like I said, I will post this online where you can go through it and read all the slides and go through it as many times as you want.